Okay, so today we'll see about the second part of molecular basis of inheritance. In the previous section, we saw about what is a genetic material and we saw some historical background of the genetic material. Now today we'll take it to the next level. We'll be seeing what is a nucleic acid and we'll be seeing what is a nucleotide. We'll see both the things. First, let us start with nucleic acid. Okay, when you're talking about nucleic acid, it is a polymer of nucleotide. And what is a nucleotide? A nucleoside plus a phosphate is called as a nucleotide. So let me draw that for you. NS represents nucleoside. Okay, NS means a nucleoside. When you add a phosphate to this nucleoside, okay, a nucleoside and a phosphate together is together called a nucleotide. Okay, and what does a nucleoside consist of? A nucleoside consists of a nitrogenous base and it consists of a sugar. If it consists of a sugar and then a nitrogen base together, then you call that to be a nucleoside. We'll see that we are having separate topics for nucleoside and nucleotides. So let me not confuse with you for that. But now you get to know they are polymers of nucleotides. So nucleic acids are polymers of nucleotides. And what do I mean by polymers? If I tell something to be a polymer, it means there is one molecule and that molecule forms a chain. Am I right? That molecule forms a chain. So n number of chains is called as a polymer. Now this polymer, what I am discussing here, the nucleic acid is a polymer of nucleotide. So it will contain a nucleoside and a phosphate and there will be many chains of this. Is it clear? When I talk about a nucleic acid, it basically consists of three different components. The first component is a pentose sugar. Penta means what? Penta means 5. So it's a 5 carbon sugar. Basically it's a 5 carbon sugar. Second is a nitrogenous base and the third one is a phosphoric acid or what I called as the phosphate. It's called as a phosphate linkage or a phosphoric acid or a free phosphate bond. It can be called in any way. There are different names to it, but for general understanding, we'll call it as phosphoric acid. So a nucleic acid basically consists of pentose sugar, nitrogenous bases, and a phosphoric acid. We'll see about each one in detail. Okay, the first one is pentose sugar. The pentose sugar is basically called as a furanose ring structure. It is called as a furanose ring structure. I think you all know what is a ring structure. Ring structure, the benzene ring structure. In the isomerism also you would have heard about the ring structures. So the ring structures are basically the ones which are like this. It will be an enclosed circular structures that will be formed in chemical compound. That is called as a ring structure. These are called as furanose ring structure. You know what is a furanose ring structure? It is a 5-membered ring. 5-membered ring means you can find 5 carbons here. You always start naming it in this way, right? 1, 2, 3, 4, 5. You always name like this only. So when you are going in this way, so you call that to be a furanose ring. Why? Because it consists of 4 carbon atoms and 1 oxygen atom. You can find here 1, 2, 3, 4. It will have 4 carbon atom here. The 5th carbon atom will be protruding from the 4th carbon atom. You will understand, in the ring structure you will not be able to find the carbon, but that 5th carbon will be protruding out from the 4th carbon and it will be attached to that. So it will have 1, 2, 3, 4 carbons that is directly in the ring and there is 1 oxygen which is also in the ring. Okay, so this is called as a furanose ring structure. All the pentose sugars are basically furanose ring structures. Is it clear, children? Now this pentose sugar is basically divided into 2 categories. The first one is a ribose sugar. The second one is the deoxyribose sugar. Ribose sugar is found in RNA and deoxyribose is found in DNA. The structures are same. If you find C, 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 C and one more C here. Here also you can find the 4 C's and 1 C that is attached to the C. Everything is same. The only difference between the 2 structures is, if you see the 3rd carbon here, the 3rd carbon is attached to OH here. And if you see the 3rd carbon here, it is also attached to OH here. Now you come to the parallel 2nd carbon. What is attached to? H. This side OH. Here H, but here H. So this O is completely lost here. You will not be finding that O there. Is it clear? What is the reason for this? And there are many diagrams representing, if you can find in the textbook also, what they'll do is they'll always try to shift these both. One O will be present here, in this diagram O will be there, and that diagram O will not be there. But basically, to identify which is an RNA and which is a DNA, you should find the second and third carbon. If in the second and third carbon, both the carbon are having OH and OH, then you can tell it is a ribose. If in the one carbon there is OH and in the other carbon there is only H, then you can tell it's a deoxyribose. This OH is a very, very important functional group. It's an alcohol group. You would have read about it. OH is an alcohol functional group. This alcohol functional group is a very, very reactive group. It's very reactive, right? So basically, if you see, how many functional groups are here in ribose sugar? 2. How many functional groups are there in deoxyribose? There is only one. Only one OH is here, 2 OH are here. So here if you find there is 2 and here there is only 1, which makes the ribose more reactive. So this makes the ribose more reactive. Since there are more number of functional groups, obviously it is more and more reactive. Since it is more reactive, it is unstable. Because once if it is constantly reacting or it is in a state of reaction, then it is purely unstable. And if something is unstable, you cannot take that as a basic chemical to work with. So that is why they always tell DNA is the most stable genetic material than the RNA. So if you take any kind of a eukaryotic organism, you will always be finding only deoxyribonucleic acid and you will not be finding a ribonucleic acid. So because of this functional groups, this ribose sugar seems to be even more reactive and highly unstable in comparison to deoxyribose. Is it clear? So next we'll be seeing about nitrogenous base. Fine.